Hi everyone, thanks for joining today's webinar. So today we'll be discussing how to use the clinical and epidemiological resource ClonipDB to explore data, and this is aimed primarily at genomics researchers, so biologists, wet lab scientists, etc. So to start, just some housekeeping rules first. You should see a panel similar to the one in this screenshot here, and you should see that you're on mute by default as a participant. But we are very happy to take questions at any time. So if you have a question, please just type it into the chat box. We've got a team of people here who will be answering questions as they come in. And then at the end of the presentation, we'll also be answering questions out loud. I also just wanted to let you know that this presentation is going to be recorded, and so that will be made available later this week to anyone who's interested. Okay, and before we really dive in, I just want to acknowledge our broader team. So pictured is the ViewPathDB team. So ClonipDB is one site that's supported by a whole group of people. Our funding for ClonipDB is primarily from the Bill and Melinda Gates Foundation, but the ViewPathDB project as a whole is funded by multiple sources shown below. And of course, we also need to acknowledge our data providers and the actual study participants whose data we're going to be looking at today. So first off, what exactly is ClonipDB? It's an open access clinical and epidemiological database resource that aims to really encourage exploration of complex studies. And so today we're going to focus mainly on walking you through the site, looking at the different features, answering some different questions that wet lab biologists might have of the data. But if you're interested in learning more about how we actually integrate data and seeing more about the features and tools, you can always check out our paper. So this is published in Gates Open Research. You can just search for that in ClonipDB. And that'll tell you more about the behind the scenes work of how we get things loaded in. So for today, we'll talk first a little bit more broadly about what types of data are actually in ClonipDB, and then I'll take you to the live site and we'll walk through some example questions and highlighting the different features of the site as we go along. And at the end, I'll tell you some of the things that we have planned to really bring together the epidemiological resource we have here with some of the other genomics resources that we support, like vector-based PlasmoDB, etc. So first, one of the goals of the ViewPathDB project as a whole and ClonipDB in addition is to really make data fair. So this stands for findable, accessible, interoperable, and reusable. And we do this in a number of different ways. So for one, to make data findable, we use unique study identifiers. So each study that we load has its own identifier that's stable over time, so you can always navigate to find that data set. We make data more accessible through a few different features of our website. So we have the search wizard for data exploration, and these are all things I'll be showing you today. Search strategies similar to what the genomic sites have for more complex queries, analysis apps to visualize the data however you choose, and the ability to download data for further analysis offline. A key underpinning of our site, and one thing that makes us different than a classic repository, is that we harmonize variables semantically using ontologies on the back end. So this means that if you're looking at a variable like sex in one study, you can be sure that it means the same thing as sex in another study, essentially. And finally, we aim to really make data reusable. So we provide study pages that provide the context and methodological information you need in order to make sure you're asking questions that make sense for that study. So at the moment, ClonipDB operates on a similar release cycle to the genomic site. So we release data approximately every 2 months and new features as well. And at the moment, there are 27 data sets that are available through our website, through our live website. And these data sets at the moment are primarily infectious disease focused. So we have a number of malaria studies, enteric disease studies. So these include everything from bacterial and viral causes of enteric disease to parasitic causes like Cryptosporidium and Giardia. We have a number of schistosomiasis studies, and we have a respiratory disease study that's in the works but not yet public. We also, going forward, are integrating a lot of maternal, newborn, and child health studies. And these, while not necessarily primarily focused on infectious disease, will also often contain infectious disease data. And so we have different foci in terms of, what types of infectious disease studies are focused on, but we also support a wide variety of study design. So as you look through the site, you'll see that some studies are observational, so studies that are, case control or longitudinal cohort studies that are just seeing what happens in the population, and others are actually experimental, so they're randomized controlled trials in ClonipDB as well. And across these different studies that are loaded, we support a wide variety of data types. So depending on the study, we load essentially what the study collected. So usually they'll have demographic information, they might have socioeconomic or household environment, like risk factors for different infectious diseases. They often have clinical observations, so temperature, things of that nature. Anthropometry, so measurements of height, weight, etc. Travel history, and of course, importantly, microbiological results. And so these different data types are just a sampling of all the different types of data we actually support on ClonipDB. And overall, at the moment, we have data on over 1 million participants, and you can see the breakdown between how many of those are in enteric disease versus malaria versus schistosomiasis studies. And a lot of these studies that we have loaded have a longitudinal component, so they'll follow the same person over time. And so we have actually over 5.5 million observations on these participants and over 3 million data on samples coming from these participants. So there's a lot of data that's currently in the site. And as I mentioned before, we're loading new studies constantly. So that number is increasing pretty much every 2 months. So for today's demo, we're going to focus on a few different questions. And I've put together some questions that I hope are potentially of interest to this community. So one example is, if you are a researcher who has a specific question that you need human samples to interrogate, one potential thing you could do is use the data in ClonipDB to find potential samples of interest and then reach out to the study team to see if they still have those samples that you can then do your own tests and analyses on. So the question we'll ask there is going to be, which blood samples came from participants with asymptomatic Plasmodium infection? And we'll get into that in just a second. Another question people might have is, we've got 2 different molecular detection methods for an organism of interest. Like, how do those 2 different methods compare? So for that, we're going to use the MALID dataset, a study of enteric disease, and compare the ELISA results to the TAC results for cryptosporidium detection. And then the last example, if we have time for it, is going to be, you might be working on a mouse model and wanting to compare the phenotype you're seeing in your model to actual human data. So in that case, we're going to be using data from the Khalifa-Burgo malaria cohort study and looking at how blood counts change due to Plasmodium infection. So I'm going to switch now to the live website. So this is just clinipdb.org. And sorry, I'm going to zoom in to make it a little bit easier to see my screen. So here's the welcome page. Normally, when you first come to the site, you'll be greeted by a pop-up box that just explains our data access and use policy, and you can find that again here in the About section. It's a good thing to just review this briefly to make sure you know what the requirements are. But going back to the home page, for the first example, we're going to be using data from the Prism study. So Prism stands for the Program for Resistance, Immunology, Surveillance, and Modeling of Malaria in Uganda. And this is a study that is part of the International Centers of Excellence for Malaria Research Program, or ICMR. So if you've never heard of the ICMRs, they're funded by the NIH, and they're centers that are all around the world that are really trying to understand malaria in different contexts. This particular group is studying malaria in Uganda, and this was actually it was actually the first data set that led to the prototyping of ClonipDB. So here, if you click on the study name, it takes you to the study page, which I mentioned earlier. And again, I would recommend for any study where you're really actually diving into the data, I would recommend taking a look at these pages to really understand the methodology of how the data was collected. So on this page, you can find publications associated with the study, and you can find a description of the methodology of how they collected the data. So here we can see their overall objectives, we can see when data was collected, And in this case, we've attached a chart that actually specifies, how households were enrolled. So in this case, they enrolled people from 3 different sites, 3 different regions of Uganda. And what they did is they randomly selected households in these different areas, would approach the households, and if they were amenable, they would enroll all the children within the household plus up to 1 adult caregiver as well. And so you can see the eligibility criteria here. And if you scroll down even further, you can find information on the case report forms that were used to collect data and the data dictionary. So this is helpful if you're really trying to dive in and understand how a variable was collected. And at the bottom, you can actually see a list of the researchers who are associated with the project. So you know who to contact if you're interested in the data. Up at the top, you'll see these icons next to search the data. And if we go back to the home page, you see these same icons at the bottom of the study card. And what these do is let you actually dive in and see all the different variables. And there's some slight nuance to how these differ. So basically, this participant icon lets you see all the different variables, but the data is calculated at the top at the level of the participant. Whereas if you do an observation level search, the data at the top is at the observation level. So one participant could have multiple observations in the study because it's a longitudinal cohort study. If you do a household search, you're going to instead see the data calculated at the household level because multiple participants could be coming from the same household. And this entomology search is a little bit different. This contains only the household and entomology collection data here. So for this example, we're going to do an observation level search because we're interested in identifying which blood samples, how many blood samples there are coming from participants with asymptomatic Plasmodium infection over the course of the study. So this is the basic search. And so you see at the top the name of the study, and you see a link, and this actually takes you back to the study page in case you need to reference that again. And here you see the steps, you see different steps, and you can jump between these by clicking forward and backwards. And it doesn't matter, you can go in between them in any order. And what these do is they categorize the variables into different groups. So under the participant step, what you're going to see is variables that tend not to change per person over time. So things like their genotype, their birth date and sex, etc. Under household, you're going to find information like, what was their floor made of, or how much standing water, how many sources of standing water did they have around their house, things of that nature. And under observation, this is where you're usually going to find the bulk of the data. So this is going to be anything that could change for a person over time. So in this study, you can see, temperature is one variable that was collected, diagnoses, signs and symptoms. So these are all different variables that you can find here, and sample, you can find the lab tests here as well. If you happen to already know that you're interested in certain key variables, you can search for them up here using this bar. So for instance, if I know I want to look at their Plasmodium variables, I can start typing in Plasmodium and return a list of all variables that contain Plasmodium in the label. And I can jump to them directly, and it doesn't matter which step they're in. But here you might notice that it pulls up CD36 genotype as well. And that's because Plasmodium is actually mentioned in the definition. So this search, it searches the labels, it searches the variable definition if there is one. So we try and provide these if it's particularly helpful for understanding the variable. And it also searches the values of the variable and also the original variable name if you happen to have the original variable names from the data set. So that can be a very useful way to find specific variables that you're interested in. So now, one thing we might want to check is, we mentioned before that this study enrolled all children from within household plus 1 adult caretaker. So if we come and look at the age at enrollment, we can see that this graph shows, a cluster of data under 10 and then a wide range of ages for adults. And so this graph, you can actually update it to make it easier to read. So here, if we mouse over, we see the exact counts and the values. So we can see that there's 2 participants who were 6.29 years old at enrollment. We don't really need that level of decimal place. So I'm going to increase the bin width to 1 and make this graph a little bit easier to see and navigate. So any variable that's continuous data will be presented as a histogram like this in ClonipDB. And you can also change the range of the x-axis and the y-axis if you need to. So now you can see quite clearly that there's a lot of participants who are under the age of 10, and then there's a very wide distribution, but much fewer participants who are adults. If we go to the variable sex, this is a categorical variable. So instead, what we see here is a table with the potential values. So the values are just female and male in this case, and we can see that 60% of all participants were female, or 848, and 573, or 40% of participants were male. And that's represented graphically here in this sideways bar chart. So you might wonder, oh, why are there more females than males in this study? Is there something specific about these different study regions where the sex ratio is so skewed, or is it for some other reason? And if we select male and go back to age and enrollment, what we can see is the data has been updated, and these counts up here have updated as well. So we've gone from a total of over 48,000 observations down to 20,000 or so observations. And here, instead of everything being pink, we now see pink and gray. And the data that's in pink is data that matches our subset or filter. So these are data belonging to males, and data in gray is everyone else. So in this case, everyone else is females. And we can see that of the children under 10, roughly half look to be males. Whereas if we look at the adult population, we see very little pink. So it looks like really most of the adults enrolled are female. And if we go back to sex, we can do that by clicking here or the original variable. If we change this to female and then look back at age and enrollment, we see that now, again, it looks like roughly half of the children enrolled were female, but most of the adults enrolled were male. So this ability to filter variables is a great way to start looking at potential associations between variables, and it can help you find quirks in how the study population was enrolled. In this case, it points out the fact that they enrolled primarily children, and the adults that they enrolled tended to be female adult caregivers. Or you can use it to look at other variables that you're interested in studying further. So I'm going to remove this filter, and I could do that by clicking this X here. And now you see my counts go back up to over 48,000. And instead, let's focus our question on, these blood samples from asymptomatic infections. Let's look specifically for blood samples from children with asymptomatic infections. So to do that, I just clicked and dragged my mouse over the range I wanted. And if I wanted to be more specific, I could use these inputs here instead. And so now we're down to 38,000 observations. So I'm going to jump to observation. Actually, we want to know if they even have the sample type that we're interested in. And in this particular example, I want access to PBMC samples. So I'm going to type sample here and see what comes up. And one of the first hits is sample type. If we click on that, what we see is a table that says there are Buffy coat samples, filter papers, PBMCs, pellet, and plasma samples. And in this column here, we see the total counts for each of those samples, whereas in this column to the left, it's updated to give me just the counts of my filters. So in this case, I filtered now on just children. So I can see that of the 5,295 total samples, 4,085 of those come from children, or 77%. And this is represented graphically here. So again, pink represents data that meets the filter that you've applied. So in this case, children, and gray represents the rest of the data. So in this case, adults. So 77% of all PBMC samples belong to children, came from children. Okay. So I then want to select these samples specifically, so I just check that, and now we've gone down to 4,085, which makes sense. But then I want to determine which of these samples actually came from participants who at the time had asymptomatic Plasmodium infection. And for that, I'm going to use this variable, malaria diagnosis and parasite status. And this is a variable that the Prism team actually derived from by combining three different variables. So they used the first variable they used was the blood smear variable, so microscopy, whether or not someone had parasitemia seen on microscopy, yes or no. The other variable that they used was LAMP, so this is another method of molecular detection. It's much more sensitive than microscopy. So did the participant sample show parasitemia by LAMP, yes or no? And then the final variable they used was looking at symptoms did they have malaria symptoms, yes or no? And so if they had a positive blood smear and symptoms, it's classified as symptomatic malaria. If they had a positive blood smear but no symptoms, it's classified as this, positive blood smear, no malaria. So this is asymptomatic. And if they had a negative blood smear but were positive by LAMP, that's another version of asymptomatic, but this is with submicroscopic infection. So these are the two groups that we would want to look at specifically for samples, PBMC samples from kids with asymptomatic infection. And now it's taken my total observations down to 1,300 observations. So I can, if I want to get the actual sample IDs and then go back to the study team to request access to them, we can go to View Observations. And here, this takes us to the results page, and this should look similar to pages you see on the genomics resources as well. And one thing I might want to check is there a big skew in the sex ratio from all those samples I just selected? And I couldn't do that in the filters because here, if I click revise the search, it takes me back. Any filters that are applied downstream, they don't get reapplied. The counts for variables upstream don't get updated based on downstream filters. So if we look back at sex, we're still going to see, these updated counts are just based on us selecting children, not on us selecting these specific samples. So one way to get around that is to add columns, and in this case, I've actually already added it. I've added the column sex, and this table is just looking at our filtered data. So if I click on this histogram icon here, I can see that there's a total of 557 samples coming from female children, and 760 coming from male children. So there's slightly more PBMC samples from male kids with asymptomatic infection than females. So the other, and then I can go here to download the data. So in this case, I would want to download the sample table. I could do it as a text or CSV file, and that would get me the sample ID so that I could approach the study team about doing a potential collaboration. If I wanted to get the actual clinical variables, I could go to this, make a custom table, and choose exactly which variables I want to download. And in a study like Prism, the data is all completely public, so you can download it with no issue. But in some of the studies we have loaded, they're classified as protected, and I'll show you this in a few minutes. And what that means is that you have to submit a data access request, and that has to be approved by the study team before you can actually download the data. Going back to the results page, the other thing you can access from here is this Analyze Results button. And this gives you the option to choose between 3 different data visualizations. So distributions, 2x2 contingency tables, and data summaries, which lets you plot data longitudinally, so over time. So say I wanted to look at, I already know that there's slightly more male samples coming from boys than girls, but say I also want to check where these are coming from in terms of geographic region. I can do the distributions app, and I can add an extra stratification to see that. So here, it just takes a second for the app to load. What I can do is, in this case, let's say we want to look at the counts for sex. And I want to point out that these apps actually work on the entire data set, not just the filtered data set. So the results page is showing you just the filtered data, but these by default work on everything. And so what I first want to do is stratify based on my search results. So that will mean if I look at the selected results, that's looking just at the children with PBMC samples that who were also asymptomatically infected. And then here I can add my second stratification, and that is going to be country or county. If I click on sub-county in Uganda, that updates, and then I can look at the plot grid. And what we see is here is the data that's selected. So obviously, there's only just over 1,000 observations that fall into my filter criteria, whereas most of the observations don't fall into my filter criteria. And So to zoom in on this a little bit more, you can play with the axes here, but it's actually a bit easier to see it, in this case, in individual plots. So I'm specifically interested in just my data that I selected for. So I changed this to selected. And now I can go through one by one and look at each subcounty. And so here I can see that there's only 12 samples coming from girls and 16 samples coming from boys from Wallakuba. If I go to Kihihi, I can see that there's actually no samples coming from Kihihi with asymptomatic infection. And if I go to Ngangara, I can see that that's where pretty much all of the samples are coming from, over 500 samples from girls and over 700 samples from boys. So these visualization apps can be a nice way to further stratify the data and really dig into a question that you're interested in. Okay, so going back to the homepage, before we go on to our next example, I just want to highlight the all studies table. So you can search for studies here, and this search, it's keyword search. So if you have specific countries or diseases or things that you're looking for, you can type that in here. And you can also look more easily at all the studies on the all studies table. And here you can add or remove columns based on what you're interested in looking at. And again, you can type and do a specific search based on specific fields here. So, the next study we're going to be looking at is an enteric disease study. So I could type enteric and return those. And so you see the list of enteric disease studies now. And then the other thing I wanted to point out while on this page is that you'll see a few different levels of study accessibility. And so public, as I mentioned before, means anyone can go in and see the data and anyone can download it. Protected means anyone can see the data on our site, but to download it, you have to submit an access request, and that has to get approved by the study team. And then another level we have is controlled, where you still have to submit a data access request to download the data, but it's automatically approved. And so this is more just so that we keep track of who's using the data and for what purpose. And then finally, pre-release means that data set isn't yet released on ClonipDB, but it's something that's coming down the pipeline. Okay, so for the next example, we'll be using MALI-D. And this, as I mentioned before, is an enteric disease study, and it followed children for up to 5 years after birth. And in this case, we're going to be doing an observation level search. And the question here, just to remind you, is how does ELISA compare to TAC in detection of cryptosporidium infection? So in this case, you can see we have over 1.8 million observations of these children. And again, you can click through and see what types of data were collected, expand all and collapse all to quickly see the variables within each section. But in this case, we know we're specifically interested in their cryptosporidium data. So if I start typing crypto, you can see that there's a ton of variables that pop up. So I'm just going to click this one, cryptosporidium by ELISA result. And so this took me to the raw data from school. If you're interested, in case you don't just work on parasites, there's also a wide variety of data on different enteric disease causing bacteria. So, different E. coli, Shigella, et cetera, different viruses like norovirus, rotavirus, and then of course, different eukaryotic organisms that can cause enteric disease, like different worms, crypto, giardia, Entamoeba, et cetera. So there's a lot. These enteric disease studies in particular tend to have a lot of different microbiological results that you can look through. But in this case, we want to compare this cryptosporidium CT value data to the ELISA results. And so you can see here that out of 1.8 million observations, they have almost 40,000 ELISA results for cryptosporidium, which is a lot. And you can see that most of the time, these ELISAs were negative. So 95% of all of these cryptosporidium ELISA tests were negative, 5% were positive, and less than 1% were completely indeterminate. So one thing I might want to do, if I look over here at the cryptosporidium CT value, I see that in this case, there's actually 42,000, so slightly more test results by TAC than by ELISA. And this, in the case of this TAC, so this stands for tachyon array card, and it's basically a multiplexed qPCR method so that you can detect multiple different organisms in the same reaction. And here, if you read the definition, you can see that CT values of 35 were negative. And actually, any CT value over 35 was changed to be 35. So there's quite a spike here. It's not zoomed in all the way. There's actually 39,000, over 39,000 negative results here. But so if I want to see specifically, which ELISA results also have CT results, I can select all of these here. So we've filtered the data down to these 39,000 ELISA results. And then I can go over here. And again, data in pink is, they have an ELISA result, and data in gray means they didn't have an ELISA result. And I'm going to update the bin width to 1 to make it easier to see. And then I'll also need to update the range. I need to zoom in on what I'm interested in. So we can see here that there's a good number of samples that were positive by CT that didn't actually have a corresponding ELISA result. But I can look at the distribution here, the min, mean, median, and max, and I can compare that. So we know it's 35 for the total. The median value is 35 and the mean is 34.57 for all samples. But if I go back here and then look just at the positive samples, we can see it's shifted down to 30, and you can see that it looks like there's more of a normal distribution now. So it looks like there's a number of samples that were positive by TAC that weren't positive by ELISA, but that's something you can dig into more through this interface. Okay, for the last example query, we're going to turn to ClonipDB, a malaria cohort study. And I just realized that I'm only showing you data from cohort studies, which wasn't my intention, but that's okay. So this is a cohort study done in this area in Mali, and it's actually still ongoing, this cohort, but we only have data currently available in ClonipDB from 2011 through 2018. And this study is primarily motivated by trying to understand the immunology going on during infection, before and after infection. And so they have a number of clinical variables, and then they, to look at the clinical phenotype that people have, and then they also have immunology data as well. So if you're a researcher working with a specific mouse model of malaria, and you've noticed something in, say, the blood cell counts in your mouse model, you might want to compare that to actual human data and see how it compares. Is it actually representative of human malaria or not? And so for this, we're going to do a participant level search. And in this case, we know we're interested in blood counts. So I'm going to come over to the observation step. Under sample, I'm going to look at lab test. And under blood test, I can see different cell counts. So we can look at the percent of eosinophils, lymphocytes, monocytes, neutrophils. And you can get a sense for the range there. But what's really interesting is, what we're particularly interested in is looking at, how do those counts look during infection versus not during infection. So for this, I'm going to look at the Plasmodium test results. And in this case, they only have 3 variables. So they've got Plasmodium counts per microliter of blood by microscopy. They've got RDT results, and they've got qPCR results. Excuse me. But it's always important to look at the total counts available. So if you look here, there's only 278 qPCR results out of 74,000 observations total. RDT, there's only 564. And so we're going to use the microscopy results because there's the most data by microscopy in this case. So here we're going to go, we're going to select any sample that had at least 1 parasite identified. So I just typed 1 to select the majority of that range. And now we can go back to the neutrophils and look at the data. And what we see is that the neutrophil counts are actually significantly right shifted in during infection compared to gray, which is when people are uninfected. And if we look at lymphocytes, we see the opposite, that they're significantly left shifted. So there's significantly fewer lymphocytes and more neutrophils during infection in this population. And if I wanted to, I could go back to the participant level. I could look at age information, and I could try and get into more of the details of, like, do kids specifically show a greater shift than adults? Any of those types of questions I have, I could explore them through this data set. Okay, and so the last 10 or so minutes of this presentation, I wanted to highlight how the data in ClonipDB actually has corresponding data sets in some of our other resources and what we plan to do there. And then for the last 10 minutes, we'll be taking questions. So here, on ClonipDB, we actually have some studies like GEMS1 case control study that has data in another one of our component sites. In the case of GEMS and MALI-D, they both have data that's on microbiome.db. So subsamples of stool samples from these studies were had microbiome analyses run, and that's been loaded into that site. So if we go to microbiomedb.org, we can find and scroll over. We can find the GEMS case control data, and then we can also find the MALI-D 2-year and MALI-D diarrheal data. And so at the moment, we know that we have, the epidemiological data in ClonipDB, and we have the microbiome data in microbiome.db. And one of the goals of our project is to really make it easier to jump between the two data sets so that you can select specific samples based on clinical characteristics and then look at their microbiomes in more depth. So that's one example of where we have data in two different resources. Another is in Vectorbase. So a lot of the isomer studies, again, these malaria research centers, have data on mosquito populations that's loaded into MapView in Vectorbase. So in this case, you can search for the names of the studies and find the data. We looked at the Prism study earlier, and then if we search for Prism, we see that there are 5 different data sets in MapView and Vectorbase for the mosquito data that corresponds to the human data that we were looking at earlier. And again, these data sets aren't directly linked right now, but we plan to create those links going forward. And so in MapView, you can look at the abundance, you can change this, look at the species of mosquitoes that were identified. These are the 3 different sites that data was coming from in that study. And so that's a nice, extra bit of data. So again, you could look at certain household characteristics from each of the different sites and then look and see if that has an impact on the different mosquito species that you're seeing in those regions. And then the other thing I wanted to point out is data that's currently in PlasmoDB. So we have some, antibody data, and that is not proteomics. So we have some protein array data that was done, again, by the International Centers of Excellence for Malaria Research. So some of this data has corresponding data in ClonipDB, so more clinical characteristics. There's this other antibody array of recent exposure to Plasmodium infection done by Helb et al. Danica is actually now a member of the ClonipDB team and is on this call. But so some of her data, again, some of the data in this data set is also represented in the Prism isomer. And then the Crompton data, I think some of this is actually also represented in the Khalifa Bugu malaria cohort study that we were looking at earlier. And so if you look, at the data itself, this allows you to look at serum antibody levels. And there's a few different clinical characteristics that are available, but there's obviously much more that's in ClonipDB. So the goal is to really make it much easier to, at the moment, you can filter on clinical characteristics, you can, get the list of participant IDs, and you can take, or sample IDs, and you can take it to one of these other sites, but we want to make it even easier than that for you to jump directly between the 2 components. So I think that's everything I wanted to highlight now, and so I think we'll go ahead and move into the questions.
So maybe just a reminder for everybody, if you have questions, please go ahead and type them in the question panel, and we will read them out loud as well. There was, Sheena, while you were, as people are typing questions, potentially, there was concern for your dog in the background. So we had to indicate that that's a fresh puppy who's eager to be by your feet continuously.
Yes, he doesn't like to be put in another room. He is okay. He has a Kong and toys. He's just complaining.
As we potentially wait, I know, David, you're on as well. Is there, was there anything else you wanted to highlight from your end?
The one thing I can do is show people what a data access request form actually looks like. So at the moment, I'm not logged into an account. Amazonia, Brazil is one study that does require a request. So if I just click download data, it tells me I have to acquire research approval, which means I first have to log in and then I have to submit the request. So if I log in here with a personal account that doesn't have access to anything, if I click again, I have to submit the data access request, and there's basically 4 questions. So the top is filled out automatically, the date, name, my email, but then I have to enter the purpose for what I want to do. If I've discussed it with the study team, this isn't a requirement. It just can help with the approval process if you've already discussed it with someone who's on the team. My main hypotheses, my analysis plan, and how I plan to actually disseminate the results going forward. So once this is submitted, each study has a slightly different approval policy. Some basically have a passive approval where we just wait a few days, and if we don't hear back from the study team, it means no one had any issues with the request. Others, they actively have 1 or 2 people reviewing every request, and so we have to get signed. They have to actually say, yes, this is okay. But usually it's a pretty short process. Within a week, you get approved and can download the data.
Just so we have, for example,
David,
had.
Just before you go on, there is a question. I want to make sure we get to that question as well before you go ahead. Or do you want to go ahead and say something about this specifically?
No, that's fine. I can read out this question from a researcher working on Candida infections who was wondering whether we're expecting to have any data from this type of infection, and I can respond to that and say that yes, we certainly recognize the importance of a wide variety of infectious infectious disease problems, particularly in the field of fungal pathogenesis and areas such as the Candida auris outbreaks that are causing so many problems. As you'll note, looking through the studies, there are no fungal data sets that are currently in process. If in what Sheena is showing now, if you were to type in fungi there or Candida, you wouldn't see anything coming up. This is something that has been flagged up as a priority for relevant data sets, and we've discussed with various investigators in the UK and the US. If you have particular data sets that you think would be appropriate for ClonipDB and would benefit the broader user community, feel free to contact us. As with any questions you may want to ask, the common site for any questions you might want to ask are to click on the Contact Us link at the top of any page. More generally, let me just address the issue of available studies.
First, I just wanted to say, I think we do have 1 or 2 studies that have Candida, and they have microbiological information on Candida. I don't know, I think one of them is perch, which is not yet
exactly
released.
I was going to say, yes, the perch study, which is a respiratory infection study, does have variable outcomes, I believe, that include information on Candida. It's not fundamentally a study on candidiasis. But certainly, if there are particular studies that you're interested in, Pilar, that you would like to suggest, we would be happy to hear from you about that. This is a growth area, and we're actively working on prioritizing and loading data sets. Two other comments that I wanted to make with respect to what data is available and how and when that becomes available. We have a long history in the various genomics resources we've worked with in building trust with the community to ensure that data is not released unless and until it is ready to go public. But we do often work with data providers well in advance, sometimes years in advance of data release. As you've seen, the data access that most studies are accessible for exploration on the site, even if they cannot be downloaded. And downloading typically requires a pretty straightforward form for data access. There have been well over 100 requests to date for data access, and virtually all of them have been approved, occasionally with requests for a little bit further description of the analytical plans for that data. Finally, should mention that we are also working on, but do not anticipate releasing for a number of months, a prototype study that will allow users to upload their own data for their own private analysis using a consistent platform like this that they can share with colleagues or with journal reviewers, or when the time is right, on the public ClonipDB website. If you'd like more information on that as it emerges, again, click on the Contact Us link and we'll be happy to keep you updated.
Yeah, so there's a number of other questions that came in about data that's available. So one person asked if there's any studies on Chagas disease, and sadly, no. I did my PhD on Chagas, so I hope that we get some Chagas disease studies soon. Again, if you have studies in mind that would be a good fit, you can always contact us and we'd be happy to look into loading those. Someone else asked about viral disease data, and for the most part, so the different diarrheal disease studies all have virus data as well. So the GEM studies, MollyD, Provide, they all have a lot of different viral data, particularly viruses that are known to cause diarrheal disease, obviously. Perch, once it comes out, will also have viral disease data, but those viruses are obviously more the respiratory viruses. And then some of the other studies do contain viral data as well. So in particular, the India fever surveillance study was actually a study looking at non-malarial causes of fever in one region in India. So they have data on dengue and chikungunya and a few other major viruses there. So you really have to, I mean, the best way to find out about specific studies of interest at the moment is either just searching for them yourselves, or if you have specific questions, you can contact us because we know the contents of the different studies. One of the things we're going to develop is the ability to search for variables across studies to make that easier for people to find data that they're interested in. Then another question is on using the microbiome data, can one do the split analysis? So looking at pathogens versus no pathogens in the background of commensals. I am not sure. I haven't spent as much time with microbiome DB. David, Omar, do you know?
So I guess I'm not quite sure that I understand the question. So I think what you're asking for is to look at generate a contingency table, or do a subsetting analysis on pathogens versus no pathogens. We've shown the question is, are you talking about, let's say, some sort of a components analysis that would allow you to remove the impact of commensals? Certainly the analysis of microbiome data that is available through MicrobiomeDB does display commensals as well as pathogens. And in MicrobiomeDB, you can conduct your alpha or beta diversity analyses at any level of phylogenetic resolution that you're interested in, the species level or the genus level. There is no tag to explicitly highlight pathogens versus non-pathogens, for the simple reason that, of course, pathogenesis depends on context. I'm not sure if that provides an adequate answer. If you have additional questions, perhaps you can type them into the chat.
Mm-hmm. And then someone else was asking if there's any issues with using the database to perform searches based on studies uploaded to ClonipDB for teaching undergrads. And I would say there's no issues. So if you're going to teach undergrads, I would just make sure that they take a look at the data access and use policy. The biggest thing is that we don't want people attempting to re-identify participants. So that's a major thing. The other thing is if you end up, if they end up presenting this work to others, or even actually publishing on it, then they should, they need to recognize the contributions of the original study team, presenters, etc. But if you just take a look at this access and use agreement, I think it's perfectly fine to use the data here. And in fact, we would encourage it in terms of classwork.
One thing that's worth pointing out, Sheena, is the workshop pages or exercise pages that are available. And so that's something for anybody who's would like to use the site, this one or any of the ViewPathDB sites for instructional purposes, that actually we do have a number of instructors who do use, for example, PlasmoDB or TriTripDB in their teaching. And we have a lot of material available that you are welcome to use, but from YouTube tutorials to actual exercises. So Sheena is showing you here a recent workshop that was conducted before the ASTMH meeting. And that includes, in-depth exercises, step-by-step walkthroughs that you're welcome to use, modify. And if you end up modifying them into something better, then feel free to share with us, because we'd be happy to take advantage of your work as well and make it available to our community.
Yeah, with respect to this particular question that focused on general parasitology courses for undergraduates, there are many hundreds of institutions that have taken advantage of the tutorials that we've provided in parasitology on our genomics sites, on the ViewPathDB parent database, that are specific to or that focus on examples from parasitology. Certainly we here have used many of these exercises for teaching parasitology to undergraduates. The specific question related to issues about patient data in ClonipDB, and as Sheena has already indicated, many of these studies are publicly available and there are no restrictions on that use, but that does not obviously that does not remove the general obligation for appropriate data use and description. Although these are all de-identified data, the identification of subjects is a moving target, and it would be inappropriate to try to re-identify for data sets that you would like to be able to download for your students to work with offline. That requires filling out a data analysis request, and in that form we'd expect you to indicate what the intended use is, and it's up to the study providers as to the way whether they would like to see their data used in a classroom setting. In general, the answer is likely to be yes, but it does depend on the desires of the data providers.
Yeah, and a lot of the questions you might have with data can be answered on the site without even downloading the data, and then you don't need a data access request. So I just wanted to point out, we have information under workshops and then also under tutorials and resources. So you can find, video tutorials here and written tutorials, past workshop exercises here, and then more broadly, global health data resources. So you can take a look to see if any of these are also potentially helpful for you in your work. And then another question is, what's the rationale behind the studies you accept? David, do you want to take that one?
Sure. So, so there's a mixed rationale behind the studies that are accepted for data loading. There are a variety of factors that go into these considerations. First and foremost is utility to the epidemiology research community. Our goal is to try to expedite discovery and translational research, and so we probably many people on this call have been involved in studies that where all of the most sensible analysis is reported in a publication form, and there isn't a lot more information to be extracted from that data. That would not be a high priority for data loading. There are many other studies. For example, Sheena, some of the ones that Sheena showed, the MALED study is a phenomenal study with daily records of many hundreds of individuals in countries around the world. And despite the hundreds of publications that have taken advantage of that data, there is vastly more information that one might want to extract from it. So that would be a high priority. A second criteria for consideration is the ability to get synergistic information from the studies that we have loaded. So, for example, the fact that there are a number of malaria studies, a number of enteric disease studies that are available within ClonipDB argues for the disproportionate value of adding additional enteric and malaria studies. In addition, on our side, if there are thousands of variables that need to be defined in a rigorous ontological framework, that's a very labor and cost intensive exercise. And when we have already dealt with 1,900 out of 2,000 variables from previous studies, it makes it much more cost effective to move forward. And finally, of course, there is the interest of stakeholders who have been interested in supporting this project. Funds have come from the Bill and Melinda Gates Foundation, the NIH, the Wellcome Trust, various other organizations in different ways and forms. And their interests are, of course, part of the consideration. But all of these funding agencies we've been pleased to recognize have been very forward-looking in recognizing that the value is in the information that they provide, not whether that information comes from studies that they happen to have funded.
Thanks, David. So other questions or comments? Mala, commented on a study we might want to look at for Chagas disease from Robert Gilman's lab group. And then someone else was asking if this platform is working with collecting Toxoplasma gondii data. And at the moment, we don't have any toxo data in our resource. And I'm not actually sure that any of the studies we're currently working on have any toxo data either. Again, like David said,
I do not, I do not believe so. I'm not sure if there have been any secondary studies on any of the projects that relate to cognitive impairment. For example, in the MALED study on Toxoplasma infection. In response to the question about Chagas data, and on a related note, with other Cnidoplastid data, particularly from some of the large-scale studies on Leishmania data, perhaps that's a question that we can take up offline if you'd like to contact me separately. This is a topic that has come up on several recent occasions.
Yeah. So we're at the top of the hour now, and it looks like questions have pretty much stopped. So I just want to say in the last few seconds that if you're using ClonipDB and ever have any questions, please feel free to use the Contact Us link, and one of us will respond to you and help you with whatever it is you're trying to do. And we look forward to hopefully hearing from you. And any last thoughts, David?
I don't think so. I mean, only to indicate that one of the many things that you can ask in those comments is if you are, for example, working with others in your research group or in your institution or in a course that you're teaching and would like some assistance with that, We're certainly happy in these days of COVID, when we're all very comfortable operating on Zoom, to run or GoToMeeting, to run webinars or open discussions with your research group at whatever time may be appropriate wherever you sit in the world.
Yep, very happy to do those. So thanks for your time today, and I hope you have a great rest of your day. Bye, everyone.